Dear friends and my dear students, I am Dr. P.K. Verma. In the last session of biochemistry, I had discussed enzyme structure and classification. Today, I would discuss the mechanism of enzyme action, how it catalyzes the complex reactions at body temperature, and also discuss the kinetics, including rate of enzymatic reaction and enzyme inhibition. You have studied that enzymes are the catalyst of biological system that perform various chemical reactions specifically at the body temperature without being affected by other chemical reactions taking place in the same system. When Louis Pasteur in 1860 discovered fermentation in sugar solution by yeast, Kuhnhe used the term enzyme, that means inside yeast. Kuhnhe hypothesized that the biocatalyst must be present in the cell of yeast, and these catalysts have converted sugar solution into ethanol. Sumner isolated enzyme urease in pure form from the jack bean meal Canavalia ensiformis. After that, many more enzymes were isolated and over 5,000 enzymes are known to us. It was found that all these are made of protein. Thus, in principle, enzymes are made of proteins except the ribozymes. Enzymes may be made of pure protein, but most of them are associated with some non-protein substances known as prosthetic groups. The protein part of enzyme in this case is called apoenzyme, and the whole structure is called holoenzyme that includes apoenzyme and the prosthetic groups. In the enzyme, every protein subunit has a specific tertiary structure that makes specific folds and clefts or grooves on the surface. These clefts have definite size, shape, amino acid sequence, and arrangement of alkyl side groups of the constituent amino acid present therein. Generally, 5 to 7 amino acids in these groups are important in chemical reactions, and others help in proper orientation of the reactive group and reactants. The size and shape of the cleft determines which molecule of reactant, now called substrate, can diffuse into it and make proper bonds with the alkyl groups. In this way, the structure decides the substrate selection with very little confusion in the molecular structure and properties. These clefts are known as reaction centers. So you have studied the tertiary structure of protein. You have studied that due to formation of hydrophobic bonds and ionic interaction in addition to the hydrogen bonds and peptide bonds. The tertiary structure forms a three-dimensional structure. It has numerous clefts, larger and smaller on the surface. In the larger clefts, there are 6 or 7 amino acids or sometimes 10 amino acids that have specific alkyl groups exposed to the surface of the cleft. So when a molecule diffuses into it, if it is very small, it will make a few bonds. But if it is just smaller than the size of the cleft, it will make maximum bond with the alkyl groups of amino acids present in the cleft. So, shape of these clefts, amino acid sequence and arrangement in this cleft determines to which substrate they will bind properly. And in this way, they select the substrate for the reaction. And therefore, these are called reaction centers. These reaction centers at which the main reactant is bound non-covalently are known as active sites or active centers of the enzyme. Because the reactants bind there for a short period and then after producing the products, these are removed. So the bond should be weaker and non-covalent. These specific sites at which the main reactant reacts, main reactant becomes bound is known as active site or active center. There are some sites at which an organic or inorganic substance can be attached and help in the reaction. These centers are known as allosteric sites. The substances that increase enzymatic activity by their attachment to the allosteric sites are known as activators. Inorganic substances may become bound more strongly and are known as cofactors. Attachment of these cofactors enables the structure of enzyme proper fit for the reactant. For example, the salivary amylase acts properly in presence of chloride ion and the malate dehydrogenase reacts properly in presence of magnesium ion. So these magnesium ions and chlorides act as cofactors. The organic substances mostly become bound non-covalently and are known as coenzymes. Coenzymes are normally known to us in the name of vitamins. There are some metallic ions, some fluorides and organic compounds such as organophosphates that can be bound to allosteric site or other places in the enzymes, disturb the structure and function of enzyme temporarily or permanently. These are called inhibitor substances. The inhibitors are metabolic poisons because in their presence enzyme activity decreases. The proper enzyme either cannot react or react very poorly. So these are inhibitor substances. Now let us revise how the enzyme acts. The first model of enzyme activity was given by Emil Fischer in 1860. He gave the model known as key and lock model. He suggested that the enzymes are very specific to their reactants called substrate as well as the products. It is because the enzymes and substrates have a relationship analogous to key and lock. The substrate also has a definite structure, so the reaction center also should have a definite structure so that they can bind with each other. That is, the substrate fit well inside the active sites, and on this basis these are identified. In this diagram you see that on the surface of enzymes there are two active sites. These two sites are proper fit of the same shape as the shape of the surface of two substrates, and therefore they bind. The reactive groups of these two substances, shown in yellow and blue colors, come in contact of each other without any change in the structure of enzyme, and after that the reaction is completed and the product is removed. So it is the property of the enzyme to bring the other reactants also for the reaction in the proper manner. However, if the structure of enzyme would be as rigid as keys, it would be difficult to explain how the other reactant is able to be attached or react in proper way. Practically, it is seen that at first one substrate comes to enzyme, then another substrate comes to enzyme. It means attachment of second substrate is possible only after attachment of the first substrate. If the structure is rigid, either of the two can be attached on active site at first, A or B. But practically, when A is attached, only then B will be attached. So, the rigid structure of enzyme cannot explain why there is a serial attachment of the substrates. To explain it, Daniel Koshland in 1958 suggested induced fit model. He suggested that the structure of enzyme changes at different pH and ionic strength, and the structure of active sites are not rigid. Attachment of cofactors and coenzymes make the structure of enzyme proper fit. To identify the substrate, and attachment of one substrate may further modify the structure to identify another substrate. That is, after the first substrate is attached to one active site, another active site becomes proper fit to identify the second substrate. So there must be a change in enzyme structure at the time of reaction. Thus, at every step, the enzyme undergoes structural changes to carry the reaction. So the hypothesis is known as induced fit model of enzyme activity. Similarly, when a more complex substrate becomes attached, the structure changes in a way that it breaks the bonds between the two constituent units of this complex molecule. When the products are released, the tension on enzyme also is released and enzyme returns to its original configuration. Let us see this another diagram. You can see the structure of enzyme is not perfectly fit to identify the two substrates. But when the substrates come to enzyme, the grooves become changed and accept the two substrates. After the bond is formed, again enzymes come to its original shape. In second diagram, you can see that the substrate is a complex molecule made of two units. When it binds the enzyme, the size of cleft, the shape of cleft slightly changes to become proper fit to the complex substrate. After that, The structure of enzyme changes a bit and the bond between two subunits break, and after breakage the two parts of the subproducts are released. Again the enzyme comes to the original shape. So there is slight changes in the reaction centers in the enzymes during reaction. Now the question is, like other catalytic reactions, enzymes increase the rate of reaction by lowering its activation energy. Some enzymes can control conversion of substrate to product many millions of times faster. There is an extreme example of ornithine 5' phosphate decarboxylase. It allows a reaction in milliseconds that would otherwise take millions of years to occur. That is, the reaction normally does not take place in nature, but in presence of this enzyme ornithine 5-phosphate decarboxylase, the reaction occurs in milliseconds. It means the rate of reaction is increased millions of times. In our body, carbon dioxide dissolves in water to form bicarbonate ions, but the rate of reaction is very slow. In presence of enzyme carbonic dehydrogenase, conversion of CO2 into bicarbonate becomes very fast, and almost all carbon dioxide produced in our body convert into bicarbonate ion in the RBC and also at the epithelial surface of the lungs and gills. So these enzymes increase rate of reaction hundreds times, millions times more than the normal rate of reactions found at the normal temperature. Enzymes lower activation energy in following ways. Number 1. Enzymatic reaction take place in more than one intermediate reactions, specifically involving the following step. At first, enzyme binds with substrate to form enzyme substrate complex. Now the substrate changes into an intermediate compound forming enzyme intermediate complex. Now the intermediate complex changes into product and the enzyme product complex is formed. Lastly, the enzyme and product separate. So at least there are 3 steps in enzymatic reaction: ES complex, EI complex and EP complex. At each stage, only a smaller part of energy is needed. In broad sense, you see, without enzyme, the energy needed is very high. It is shown in red color. The example that I just had mentioned about the carbonic anhydrase, carbon dioxide dissolves in water forming H2CO3 and the H2CO3 breaks to form H plus and HCO3 minus. After presence of the enzyme, the energy level decreases, the activation energy is very lower and the products H2CO3 is formed. It is simplest reaction. Now, when the enzyme substrate, enzyme intermediate, enzyme products, these 3 steps are performed, the required amount of energy at each stage is E1, E2, E3. The total energy of reaction is same, but it breaks into three fractions. When the second substrate becomes attached to enzyme, it comes into higher energy status and its structure changes so much that the reactive groups of the two substrates are brought together in the bond forming distance. Such change is called orbital steering. Let one substrate is A, another is B. These two will be picked up by the enzyme in a way that their reactive groups are outside the reaction center. After changing the structure of enzyme, the reactive groups of both the molecules will be brought together by the mechanism known as orbital steering. It saves so much energy which is wasted during random collision of the molecules during simple chemical reaction. In a simple chemical reaction, when you increase the temperature, the movement of the molecules increases, their kinetic energy increases, and they collide each other randomly from various angles. Let us take an example. If the two molecules had cubical size, cubical shape, they have 6 faces each. So the possible collision is 6 into 6, 36. If one has 5 faces, another has 6 faces, the possible collision are 35 types. 30 types, 6 into 5, 30 types. But only one collision is capable of making the bond. So energy is wasted so much in making another collisions. 1 out of 30 collisions or 1 out of 36 collisions is capable of forming the bond. The other collisions are useless and nothing but wastage of energy. So it saves so much energy which is wasted during random collision of the molecule during simple chemical reaction. It means that enzymatic reactions have tandem collision, not random, it is tandem collision. The higher energy level is needed only to increase the rate of collision. The actual amount needed to form the bond is much lower. The enzymes, instead of random collision, perform a tandem collision between the reactive groups, and therefore by expenditure of minimum energy bond, minimum energy is formed. When a larger molecule is attached to an enzyme, it needs a larger group which is formed by utilizing two different domains. Once the attachment is completed, the structure of enzyme changes and the substrate molecules break into two parts. Energy is lost and the two domains move away. Enzyme-like aldolase functions in this manner. It forms dihydroxyacetone and glyceraldehyde. Now, what are the potential mechanisms for enzyme catalytic efficiency? The first is by binding to the substrate at the active site, enzymes can increase the effective local concentration of reactant. That is called proximity effect. Substrate binding can correctly orient the reactive groups at binding site, that is called orbital steering. Enzyme can promote desolvation upon substrate binding. Enzymes can enhance the inherent reactivity of functional groups by altering the microenvironment in the active site. Now, when you have to measure the enzyme kinetic, you have to measure the rate of reaction. Let us assume that the reactant is R and it produces two products A and B. Like any other chemical reaction, the rate of enzymatic reaction also is proportional to the molar activity of reactants or the products of molar activities of reactants. The reversible reaction follows the rule of chemical equilibrium. You must have studied law of mass action. In that case, you have studied that the rate of reaction is proportional to the molar activity of the reactant. The rate of reaction will be proportional to the product of the product concentration, and the reverse reaction, forward reaction, is the proportional to the molar activity of substrate. And when these two rate of reactions R1 and R2 are compared, there is a constant K that would be equal to product of A and B divided by the substrate AB. When the given amount of enzyme or enzyme extract is taken, the activity increases by increasing substrate concentration. However, after some time, the rate of reaction becomes constant because all the molecules of enzymes are utilized, are saturated in performing the reaction. If you will increase the amount of enzyme, rate of reaction will increase. If you will decrease the amount of enzyme, rate of reaction will decrease, and therefore a definite amount of enzyme should be taken. The highest activity of enzyme in this condition is called maximum velocity or Vmax. For highly purified enzymes, it may be constant in terms of per milligram, per minute or per hour, but practically the activity is recorded in tissues or in blood plasma, etc., and the values change depending on the actual concentration of enzymes in this sample. So when calculation is made, it is made in terms of milligram tissue or milliliter blood plasma. It is directly proportional to the concentration of enzyme in that sample. Therefore, Vmax does not show any standard property of any enzyme. It is variable. Michaelis and Menten in 1913 studied a number of enzymes, made graphs, and he found that the substrate concentration at which the activity of enzyme is half the maximum velocity is a constant for any given enzyme. That means you can change the amount of enzyme changing the value of Vmax. But if you will consider half velocity, the amount of substrate is constant whether the actual amount of enzyme is higher or lower. This constant has no effect. So this was named as Michaelis-Menten constant or Km value. Here a constant amount of enzyme is taken. The concentration of substrate is increased gradually to achieve Vmax. When the velocity of reaction was half on the baseline, the amount of substrate was calculated and it was called Km. If enzyme is absent, the reaction will be linear. There are many chemicals in presence of which the rate of enzymatic reaction decreases. These chemicals are known as inhibitor substances. There are two categories of such inhibitors, the reversible and non-reversible inhibitors. In case of reversible inhibitors, the activity of enzyme is recovered by decreasing concentration of the inhibitor substance, so the damage due to inhibitor is temporary. In case of non-reversible inhibitor, the enzyme activity is permanently damaged because the inhibitor binds the enzyme permanently. For example, most of the pesticides and higher metals like cadmium, lead and arsenic, etc. affect enzyme activity permanently. In presence of methanol in higher concentration, oxidation of ethanol into acetaldehyde is affected and formaldehyde is produced, that is toxic. But when the molar concentration of ethanol is increased, formaldehyde production becomes poor and acetaldehyde is formed. So here methanol is a reversible inhibitor for the enzyme alcohol dehydrogenase. Now there are another classification particularly for the reversible inhibitors. It is the competitive and non-competitive inhibitors. A structure of a competitive inhibitor is so much similar to the substrate. So it can diffuse into the active site, become bound therein, and can be metabolized whenever it is present in higher concentration. That means the substrate and inhibitor both have similar structure, and so their attachment to the substrate depends directly on the molar concentration. That is, if inhibitor is in higher concentration, the original reaction will be inhibited. If the molar concentration of original substrate is higher, effect of inhibitor will be inhibited. Methanol and ethylene glycol compete with ethanol for alcohol dehydrogenase. Malonate competes with succinate for the enzyme succinate dehydrogenase. and forms enzyme inhibitor complex rather than enzyme substrate complex. It produces nothing because malonate cannot be dehydrogenated. But the activity of enzyme is inhibited. It no longer can oxidize succinate into fumarate. The non-competitive inhibitors do not compete with substrate to occupy active site. They become bound to some other site of the enzyme known as allosteric site and disturb the structure of the active site, and therefore the original substrate cannot bind at the active site. The compounds like phosphoric acid and hydride, carbamic acid esters, and their mixed anhydride commonly known as diisopropyl fluorophosphate DFP is reversibly bind to sterile group of hydrolases, the digestive enzymes and acetylcholine esterases. So DFP diisopropyl fluorophosphate is a non-competitive inhibitor for the enzyme hydrolase and acetylcholine esterase. Malathion and phenylmethyl sulfonyl fluoride PMSF used in pesticides are non-reversible and non-competitive inhibitors. Sometimes the end products accumulate in the system and they act as feedback inhibitors. They bind at the allosteric site non-competitively and prevent the forward reaction, but their effect is reversible. Effect of cyanide over cytochrome oxidase also is reversible in laboratory condition. Potassium cyanide is one of the most poisonous compound that kills the animals and plants, and therefore it is said that these are potent poison that can kill the organism in a few minutes, and people think that the effect is irreversible, but in laboratory condition it can be removed properly and the effect is reverted. So it is in fact a reversible inhibitor. Competitive inhibitors do not affect Vmax, but Km value is increased. Mind it, competitive inhibitors do not affect Vmax, but the Km value is increased. However, the non-competitive inhibitors decrease Vmax and Km is not affected. The two types of inhibitors have two different effects. Competitive inhibitors do not affect Vmax, but Km value is increased. However, the non-competitive reversible inhibitors decrease Vmax and Km is not affected. These have been shown graphically in the later slides. Here there are two inhibitors. The competitive inhibitors bind at the same active site, so substrate is not able to bind there and rate of original reaction decreases. In non-competitive inhibitor, the inhibitor binds at allosteric site, affects the structure of active site and therefore substrate cannot bind. Now there are two graphs. Normal activity is shown in purple color when competitive inhibitor is used, Vmax is same, shown in green color, and when non-competitive inhibitor is used, Vmax is decreased and shown in blue color. Now what will be their effects on the Km value? It is the normal enzymatic reaction. When non-competitive inhibitor acts, the Km value remains same. But when the competitive inhibitor acts, the Km value increases. This is the purple line showing normal enzyme activity. Km is the substrate concentration shown at the base. Another one is the non-competitive inhibitor. Velocity of reaction Vmax is decreased, so half the velocity is calculated here, Km remains same. In case of competitive inhibitor, Vmax is same but Km value increases. Now, when a linear graph is plotted, that is known as Line-Weaver plot for enzyme activity in presence of competitive inhibitor, you can see that the value of Km changes. 1 by Vmax is constant, that means Vmax is constant. When non-competitive inhibitor is used, you can see the Vmax changes, but Km value remains same. So you have studied a structure of enzyme, mechanism of enzyme action, effect of inhibitors, how the values of enzyme activity and Km is calculated. In the next session, we shall study carbohydrate. See you in the next session. Thank you. Thank you very much.